God morgon och välkomna allesammans till Karolinska Institutet och dagens presskonferens. Det är ju dags att tillkännage årets Nobelpris i fysiologi eller medicin, och som vanligt kommer vi att ge merparten av presentationen på engelska, men det går förstås bra att ställa frågor även på svenska. A warm welcome to Karolinska Institute and today's press conference. The Nobel Assembly has concluded its meeting, and we are ready to announce this year's Nobel Prize in Physiology or Medicine. I am Göran Hansson, Secretary of the Nobel Assembly, and with me are Professor Julien Sirath, Chair of the Nobel Committee, and Professor John Inge Henter of the Nobel Assembly. I will start by reading the announcement in 5 languages. We'll distribute a press release. Professor Sirath will introduce the science behind the prize, and we'll take questions. And after that, there will be opportunities for interviews with Nobel Committee members for those of you who have requested that. Ready? Nobelförsamlingen vid Karolinska institutet har idag beslutat att 2013 års Nobelpris i fysiologi eller medicin gemensamt tilldelas James Rothman, Randy Schekman och Thomas Südhof för deras upptäckter rörande maskineriet som reglerar vesikeltrafik, ett viktigt transportsystem i våra celler. The Nobel Assembly at Karolinska Institute has today decided to award the 2013 Nobel Prize in Physiology or Medicine jointly to James Rothman, Randy Schekman, and Thomas Südhof for their discoveries of machinery regulating vesicle traffic, a major transport system in our cells. Die Nobelversammlung am Karolinska Institut hat heute beschlossen, den Nobelpreis für Physiologie oder Medizin für das Jahr 2013 gemeinsam an James Rothman, Randy Schekman und Thomas Südhof für ihre Entdeckungen betreffend die Maschinerie, die den Vesikelverkehr regelt, ein wichtiges Transportsystem in unseren Zellen, zu verleihen. L'Assemblée Nobel de l'Institut Karolinska a décidé ce jour d'attribuer le prix Nobel de physiologie ou de médecine 2013 conjointement à James Rothman, Randy Schekman et Thomas Südhof pour leur découverte relative aux mécanismes régissant le trafic vésiculaire, un système de transport essentiel de nos cellules. Nobelevskая ассамблея при Каролинском институте решила сегодня присудить Нобелевскую премию по физиологии или медицине равно Джеймс Rothman, Randy Schekman и Thomas Südhof за открытие в области регулирования движения пузырьков в основной транспортной системе клетках. Thank you. With that, I'd like to invite Professor Julien Sirath to present the new Nobel laureates and their discoveries. Julien, please.
Thank you, Göran. Good morning, everyone. So here are the 2013 Nobel laureates in physiology or medicine. On the left is Professor James Rothman from Yale University, New Haven, Connecticut, USA. In the center is Professor Randy Schekman, University of California, Berkeley, USA. And on the right is Professor Thomas Südhof, Stanford University School of Medicine, Palo Alto, California, USA. Over the next few minutes, I'll give you some background information about their discoveries. The 2013 Nobel laureates have been interested in questions related to fundamental cell physiology. This slide shows a schematic representation of a cell. This cell could be any cell from the body. One of the greatest mysteries of cell physiology was how the right substances could be delivered to the right destinations at the right time. How are molecules such as hormones, transport proteins, or neurotransmitters correctly routed to their appropriate destination? And how is this process controlled with temporal precision? The 2013 Nobel Prize honors 3 scientists who have solved the mystery of how the cell organizes its transport system. Each cell of the body has a complex organization that separates specific cellular functions into compartments. This compartmentalization vastly improves the efficiency of many cellular functions, and it prevents potentially dangerous molecules from roaming freely within the cell. Molecules are transported around the cell in small bubble-like vesicles shown here in this slide in blue. These vesicles shuttle their cargo between different compartments in the cell. This inset zooms in on vesicles that have been routed to the cell membrane. It shows a vesicle docking and fusing with the membrane so it can deliver its cargo to the outside of the cell. The 3 Nobel laureates have discovered the molecular principles that govern how this molecular cargo is delivered with precision to the right place at the right time in the cell. Location and timing are everything. Randy Schekman studied how the cell organizes its transport system. In the 1970s, he studied the genetic basis for this transport process using yeast as a cell model. This slide depicts vesicle transport in normal yeast cells on the left and in mutant yeast cells on the right. Schekman identified yeast cells with defective transport machinery. The blue bubble-like vesicles piled up in certain parts of the cell. Schekman found that the cause of this congestion was genetic, and he went on to identify the genes that mediate specific steps in vesicle transport. James Rothman also studied the nature of the cell's transport system. This slide shows a vesicle at 3 different stages in the transport process. It also shows the membrane of the cell. In the 1980s and 1990s, Rothman discovered specific proteins pictured here on the vesicle in orange and the membrane in orange as well. These specific proteins form a complex enabling vesicles to fuse with their target membranes, shown here in the center. In the fusion process, proteins on the vesicle and target membranes bind to each other like 2 sides of a zipper. This ensures that the vesicle fuses at the right location and that the cargo molecules can be delivered to the correct destination. But questions still lingered. How is the release of the cargo controlled in such a precise manner? Thomas Südhof, he was interested in how nerve cells communicate with one another in the brain and how signals instruct vesicles to release their cargo with precision. In the 1990s, he searched for calcium-sensitive proteins that control this process. He identified molecular machinery, shown here in purple, that senses calcium ions and triggers vesicle fusion. Calcium binds to this machinery and triggers the complete fusion of the vesicle with the target membrane. Südhof's discovery explained how temporal precision was achieved and how vesicles could rapidly release their cargo upon command. All right, so this slide will show an animation of the machinery regulating vesicle traffic. The vesicle and the membrane are shown. The vesicle is routed towards its membrane. Proteins on the vesicle and the membrane bind to each other like 2 sides of a zipper. the vesicle fuses with the membrane. Once the vesicle is in position, it must wait until calcium enters the cell. Calcium binds to a molecular sensor on this complex, and that explains how temporal precision is achieved and how signaling substances can be released from the vesicle on command. The vesicle transport system is critical for a variety of physiological processes, ranging from signaling in the brain to release of hormones to release of immune cytokines. Without this wonderfully precise organization, the cell would lapse into chaos. Defective vesicle transport occurs in a variety of diseases, including a number of neurological and immunological disorders, as well as in diabetes. The 2013 Nobel Laureates have discovered a fundamental process of cell physiology. Their discoveries have had a major impact to advance the understanding of the machinery regulating vesicle traffic, a major transport system in the cell. And here, once again, are the 2013 Nobel Laureates in Physiology or Medicine. Thank you for your attention.
Vi tackar Nobelkommitténs ordförande. Och nu är det dags för er att ställa frågor. Time for questions. Who would like to start? You are all stunned, it seems. Or perhaps you all had expected this.
Yeah, what did the prize winners say when you spoke?
What did they say? Well, to be honest, I have been able to reach Randy Schekman and James Rothman. And they were both delighted, surprised, happy, delighted to share with the two others in this combination. And they look forward to coming to Stockholm in December. Please.
Have the laureates been collaborating scientifically?
Har vi mikrofonen? Mikrofon. Have they been collaborating scientifically? Yes, I think Rothman and Schekman have been collaborating, right, Julie?
They started their work all independently. And it was very interesting that the work of Schekman in yeast and the work of Rothman in mammals, they were aware of each other's studies. And so that led to collaborations later. And I don't believe that they have collaborated with Südhof. So that's a complementary line of research.
Could you say something about clinical use of this research? For instance, what kind of medications have been created from this?
I think we'll ask our clinician, Jan-Inge Henter, to comment on that.
So first of all, we have to realize that this is a prize on physiology, on cell physiology. But these beautiful discoveries have importance for our understanding of the human body and obviously implications for diseases in various organs, such as the nervous system, diabetes, and immune disorders.
Could you perhaps give us an example of, I know that it's used in diagnostics, isn't it?
Well, yes, so there are a set up of implications. First of all, we can think of tetanus. Tetanus is a bacteria toxin that affects this vesicle transport and causes the death of hundreds of thousands of children each year. That's one example. We use also in immune disorders in children with high degree of inflammation. We use the analysis of vesicle transport in the diagnostics, and based on these findings, we decide treatments. So yes, they have clinical implications.
And there are, I think, genetic disturbances, rare diseases, but there are genetic disorders in this machinery. One of them causes a severe immune defect, as Dr. Henter mentioned, with hyperinflammation. Another one leads to, mainly affects the brain and leads to epilepsy and mental retardation. So there are clear medical implications. So let's see if there are any more questions. Seldom you encounter journalists so silent. Please, yes. Could you have
You said there are clear medical implications, but so has this discovery led to new medicines, or is it just a way of understanding how the disease works? Is it still early stages? In what way?
It has not led to any medicines yet, but it has led to diagnostics. But as Jan-Inge said, these discoveries are on the basic machinery. Others have applied them to understanding or developing diagnostics. Please.
What is the implication towards diabetes?
Diabetes? Julie?
So, yeah, the implication for diabetes that we know is insulin is produced in the pancreas and it's released in a calcium-sensitive manner. And so this machinery operates in the release of insulin into the bloodstream. The machinery also works in tissues that respond to insulin, the hormone that mediates sugar uptake. And so it's a fundamental process in all cells. And that has really led to a greater understanding of disease pathogenesis and prospective treatments.
Mm-hmm. Are there more questions? It's almost like lecturing to Swedish undergraduate students. But, you haven't asked the question that they always ask, will this come on the exam? Yes, please, we have a question up there.
Hello, this is Mauricio. I am a student at Karolinska. I would like to know if these researchers have done research on the DNA codes relating with the physiology of the transport system, or only it's related with the physiology of the Biden itself only.
Julie?
The major discoveries that we're awarding are on the protein machinery that mediates this fusion, targeting and fusion process in the calcium sensor. And of course, they have published many papers along different areas related to this process.
But Schekman's approach was genetic, as you heard in the presentation. He studied yeast cells with genetic defects and unraveled the machinery. Yes, up there.
Physical property includes not only endocytosis, but endocytosis as well. I mean, could you comment on this?
Well, you're correct, but the actual award that we're making today, the recognition is on the traffic process.
Mm-hmm. Please, down here, we have a question from gentleman on the second row.
You mentioned the central nervous system. Could you give an example of a disease that is connected with these mechanisms?
I think actually we mentioned the tetanus, because that is related to the nervous system and affecting the muscles. So this is associated with the nervous system. Yes.
Anything else?
Well, there is a disease called botulism, which is also related to the nervous system.
How does this work in botulism?
This is in the same way that there are bacteria that produce toxins, and they destroy this vesicle transport, and that causes potentially fatal disease, or a fatal disease. And we can actually use this toxin also to treat children that have spastic paralysis or dystonia in the muscles, to release the tension in these muscles, so it's used therapeutically as well.
Okay, do we have further questions? If not, then I think we'll close here, and there will be, as I said, opportunities for interviews for all of you who have requested interviews with Nobel Committee members, and I think Anne-Marie Dumansky will organize that. Thank you very much.
Karolinska Institute with this year's prize, Nobel Prize, I just say, because in physiology or medicine. It's just been announced. I'm standing here with Professor Julien Sirac, the chairman of the Nobel Committee. What discovery have you awarded this year?
Well, this year we've awarded a fundamental discovery in cell physiology for vesicle traffic, a major transport system in the cell.
At the press conference, I got the impression that this prize, people were a bit quiet. Have many people been waiting for this prize, do you think?
Well, people have been aware of this work, for decades. And, the work started in the 1970s, and it's sort of built throughout the years. So in some ways, I'm surprised that it wasn't, more of a obvious choice. But so it's a fundamental discovery of cell physiology, and it was not entirely easy for these investigators when they started. So Schekman, for example, did yeast genetics, and Rothman was doing biochemistry in a test tube, and Südhof was doing biochemistry and some functional studies with animal models.
Could you explain a bit about the vesicular transport system for our lay audience, just
Right.
in easy words?
So think of a cell as sort of a factory, and it needs to produce proteins. And they need to shuttle these proteins in cargo from one workstation to the next, so each protein can get a little bit better refined along the way. The fundamental discovery here is how the packages, the molecules, the cargo, is shuttled from one compartment in the cell to the other, and how these molecules can be also exported from the cell. So it's that process which we have awarded the Nobel Prize in.
So what were, if you should, the key breakthroughs that make them worthy of the Nobel Prize, those three gentlemen, what was the three steps?
The three steps. So Schekman used yeast genetics, and he was studying temperature-sensitive mutant yeast, and he could morphologically, with a microscope, see that some of the vesicles in the mutant yeast were building up in certain parts of the cell. And he understood that disturbed the traffic. And he went on to identify the genes that controlled this traffic. That was a pretty bold approach, because at the time, people didn't necessarily think that studies of yeast would be of relevance for mammals. Rothman took a really bold approach. He studied this process in a test tube. Now that approach was met with some skepticism, because people thought that this traffic would be dependent upon the proximity of the compartments with each other. Therefore, you theoretically wouldn't be able to break up the cell, because you'd need to have an intact cell. But Rothman broke up the cells and studied this in a test tube, and he could show that even in a reconstituted system, he could build up this interaction between proteins, the protein machinery, and study the traffic of these vesicles in broken cells. And Schekman, when he came on the scene, I think he recognized that calcium could be a sensor.
Südhof, do you mean?
Südhof, yes, correct, Südhof. Südhof came on the scene. He recognized that calcium could be a sensor. And he was faced with a situation where all the building blocks were unknown. And so his job was to really put functionality and understand what are the building blocks that the calcium binds to, and how does that release vesicles in a temporally controlled manner.
The will of Arthur Nobel states that the prizes should be awarded to those who have conferred the greatest benefit on mankind.
Right.
So in what way does this prize fulfill this criterion?
Well, that's true for all of the prizes. And new knowledge about how we are in the world is of great benefit for mankind. This, again, is fundamental cell physiology. So the knowledge that we're gaining from this is applicable to all cells from yeast to humans, showing how ancient this is. And what these individuals have done is they've uncovered this machinery, this mechanism, and that has allowed us to understand basic physiology, but it's also allowed us to have some insight into disease progression and prospective treatments. So it's a pretty great benefit to mankind.
Prospective treatments, could you just give an example in one area when this could be?
Right, so understanding the treatments better would allow you to focus on the machinery and potentially come up with drugs. For example, diabetes. It's a disease where insulin is not released from the pancreas appropriately. So this is controlled with also the calcium impulse. So theoretically, one could work in that area and develop pharmaceuticals to improve insulin release. In tissues like muscle, you have an uptake of sugar into those cells. Theoretically, one could work on trying to target that machinery to have a better interaction of transport proteins to take up sugar and feed the muscle with sugar, glucose. So there's a lot of examples of where this could be used.
So why was this prize awarded this year?
Yeah. Well, it's a long journey in terms of the vetting and how we work. And this one is mature, and we felt that we had a fundamental discovery that was Nobel Prize worthy, and so this is why we're here today to talk about these laureates.
Since there are a lot of young people watching right now, how would you explain the importance of this prize to say, like a grade school student?
Right. Okay, so if you can imagine that you were going to go to school and you were going to take the bus, and the bus showed up, but there was no sign on that bus to tell you where it was going to take you. All the signs on the buses disappeared. So you'd get into the bus without having any idea of where it might take you. It might take you to your school, but it might take you to your parents' workplace. So imagine that. You'd be missorted. And that's a little bit what this process is. Because of this process, the passengers, the cargo, they get to the right place, the school, at the right time, so you're not late, and you don't end up at the playground or at your parents' workplace or somewhere else where you're not supposed to be. So this whole process helps the cells sort proteins, so the information is moved to the right place.
That's an excellent metaphor. What was the state of knowledge in this field at this time?
A little bit of what I said to you before. When Schekman started, people were skeptical of the idea of using a yeast model. People were unsure that you'd be able to really even do these experiments. When Rothman started, it was incredibly bold. He broke up the cells, studied this in a test tube. People were skeptical. They didn't think it could work. Even with the Südhof work, the molecules were not on the table.
So when did the scientific community realize the significance of these findings?
Scientists, I think, are clever to catch on to what's new in the field, and people have really been actively engaged in this area for decades. This is a tough area of science. It's been really a tough problem to solve, and so many individuals have been working on different parts of it.
So if we turn our attention to the laureates, James Rothman, Randy Schekman, and Thomas Südhof. Do you know them yourself?
I don't know them at all personally.
No?
I mean, I've heard James Rothman lecture, but I don't have any interactions with them, and I'm not collaborating with them. But I'm aware of their work.
And do you have any idea? I mean, those findings that are awarded today, a long time, in the 80s, 90s. Yeah. What do they work on now?
Well, so some of the important work is even coming even today. So we have some papers that have been published in the 90s and the 2000 that are really of relevance for this work. So all of them are active scientists. All of them are completely engaged in their projects, and they're moving these questions forward today.
And personally, what makes you so enthusiastic about this prize?
Oh, I just think it's really fantastic. I mean, it's so beautiful. The organization of this traffic in the cell is just phenomenal. And the fact that cells really require this, otherwise they basically can't survive. It's just phenomenal how cells are able to move this proteins from one place to the other. So, I mean, I just think the cell biology, the cell physiology is really beautiful.
And finally, has the Nobel Committee been able to reach any of the laureates?
So there has been a telephone conversation between Göran Hansson and Randy Schekman and James Rothman, and Südhof we haven't been able to reach him, but we're working on it.
So how did they react?
Oh, very humbled, delighted, surprised. I'm sure it's a moment of pause for them.
Great. Thank you so much.
Thank you very much, Lotta.